Today we want to go through the process of how you take and submit samples to the Plant, Pest, and Diagnostic Clinic. This is especially important when you have some kind of disease or other problem which you're not sure. With the recent discovery of downy mildew in northern Ohio and cucumber crops being very susceptible to downy mildew, we want to check this field out to make sure nothing serious is going on here. So that's the purpose of why we're out here today. We are particularly concerned about our pickle and slicing cucumber crops. As we look at this slicing cucumber field here, we can definitely see something going on. But my first observation is that this really doesn't look like downy mildew. It doesn't have the yellow angular spots, and you don't see any mold on the bottom of the leaf. So we're going to take samples of this planting, especially just the leaves in this case, and take them off to the Plant, Pest, and Diagnostic Clinic to find out exactly what is going on here. I have my suspicions that this might be Alternaria, but we need to check this out. We also want to make sure that we are really not seeing downy mildew here, because if that was the case, these plants would be defoliated in a matter of a few days. So what we want to do is take representative leaf samples of the problem. We don't want really dead leaves. We don't want leaves that aren't showing any symptoms whatsoever. What we want are leaves that are a mixture of the problem somewhat severe and where we can see the problem starting to develop. In this case, we're not going to take the whole plant because the plants aren't wilting down and we don't need to really look at the root system. So today, for this problem, we're just going to take leaf samples. If we had a definite wilt problem here, we would need to take a whole plant sample so we could look at the stem and also the roots to see if there's anything going on in that part of the plant. So what I'm going to do is take some representative leaf samples, and in this case I have a paper bag. See what I'm talking about here. It's a good idea to get as many samples as you think are necessary. In this case, I'm going to probably pick about 10 or 12. And I would take them from various parts of the field so we have a good idea of the extent and what the actual problem is. Now, if it's going to be a while before you can get these to the clinic, I would hopefully have a cooler on hand. And if you're not going to be able to ship these out today, be sure to put these in the refrigerator overnight. And that should do it. I think I have enough samples here to get to the crux of this problem. This is the time of year we see a lot of foliar diseases start to develop in tomatoes, especially such things as early blight, septorial leaf spot, and also you can have bacterial spot and canker. On these plants here you can see some yellowing beginning to develop and also some spotting, which to me looks like bacterial speck or spot. So we're going to take leaf samples here like we did it in the slicing cucumbers and get them to the lab to check these out. Here you can get a better idea of the spots we're talking about. As you can see, some of these have coalesced together to form necrotic or dead areas on the leaf. There are also some necrotic areas along the edges of the leaf which we want to get checked out in the clinic. The clinic is located on the west side entrance to Cotman Hall on the Ag campus, and today we're going to get a behind-the-scenes look at what goes on in the diagnostic process. All right.
Hello. Leader spot. Underside of the leaf to see if there's anything underneath. So Bob, what we're looking at here are the leaf spots that on this cucumber sample that you've brought in, and I'm looking for fungal spores which will allow me to identify specifically what is causing this problem. Looking at it microscopically, we've examined it for bacteria diseases, and now we're looking for fungi, and right now I'm not finding any obvious fungi on these leaves. So what we're going to do is we're going to incubate the leaves overnight and see if any fungi develop that will allow me to identify it more specifically. And so there are lots of different ways to do this. One of the ways that we use in our lab is what we call a moist chamber box. And over here, we've dampened a paper towel, or are dampening a paper towel. And we'll lay some leaves in on this. And we don't want it too wet or the leaves will rot quickly. And so what we'll do is we'll lay some leaves in here, and both right side up and upside down. Re-examine them again tomorrow and see what has developed. Probably they should be off. Bob, you've brought in some tomato samples here into the C. Wayne Ellett Plant and Pest Diagnostic Clinic, and I'm going to take a look at them here. But first, let's take a look at the overall symptoms. You've told me that they're concerned about this spotting that they're seeing on the tomato leaves. Bob, what we're looking at here are these little tiny specks on this tomato leaf. And the first thing we're going to look for is evidence of fungal infection through the stereoscope, which gives us a moderate amount of magnification. And we might want to change the lighting and double check this. And I am not clearly seeing evidence of fungal infection at this point. So the next thing I'm going to do is I'm going to check for bacteria using a higher level of magnification. So the first thing I'm going to do here, Bob, is I'm going to take a small cross-section of a very tiny piece of the tissue, which just fell out on the bottom of the tray, and look for bacterial streaming out of that, which is definitive for identification of a bacterial disease, but it's not definitive for which bacterial disease. Let me see here. We are seeing some bacterial streaming from the small spots that were on the leaves, which tells us that we are looking at a bacterial disease, but it doesn't tell us exactly which one. So Bob, we've cut across another lesion on the leaf, and here's the edge of the leaf, and here in this kind of misty looking cloud is the bacteria that are streaming out of the leaf. We're looking at this on a computer screen, but I have it set up here on my microscope as well. In addition to looking at the small spots on this tomato leaf, I also noticed some lesions along the edge of the leaf. I once again took a small section of the edge of the leaf. You can see here where I've cut out a small piece where the lesion is, and have made a microscope slide, and once again we're looking for bacterial streaming. So Bob, once again we're looking at a different tomato sample, and what we're seeing here on my microscope over here, and I'm going to have to adjust it just a little bit. Once again, a cloud of bacteria oozing out of a cut piece of leaf. And so when we plant pathologists are talking about bacterial ooze, this is what we're talking about here. And I'm sorry it keeps darkening a little bit. Let me just lighten that up a little bit for you. As you stand and watch this, very slowly you can see bacteria streaming out of this cut lesion. So Bob, one of the concerns we had regarding the earlier tomato sample was whether or not this was bacterial canker, Clavibacter. And so we used a what's called a serological test to test for the presence of the bacteria in smashed up leaf tissue. And what we see here is a positive result. We have one band which is turned a kind of a pinkish color, which indicates that this is the control. This tells us the test is working properly. But here is the reaction to the bacteria that were in the sample. And the fact that this reaction has occurred tells us that there is evidence for the presence of bacterial canker in that tomato sample that we looked at. So Bob, I've laid over on this side a negative test result so that you can see the difference. Here again is the control line, but in this case there was no reaction to the presence of bacteria, which tells us that this sample was negative. Tests like this are available commercially, and any grower can buy this kind of testing material and test their own plants in the field should they so desire.